गुड मॉर्निंग एलेक्स। मॉर्निंग गुडिंग। सोए नहीं थे क्या अभी तक? गुड मॉर्निंग बुकी। क्यों आज थोड़ा लेट उठे? तुम तो 2:30 तक ही पढ़ते थे ना। फाइव आवर ही सोओगे शायद तुम। हाँ तो 5 आवर में हो जाएगा तुम्हारा। आई थिंक ट्रैवल में कुछ था। आई थिंक मेरा कॉफ़ी ही ख़त्म हो गया। The experimental proof. It is now proven that a DNA replicates semi-conservatively. It was first shown in E. coli and subsequently in higher organisms such as plant and human cells. Matthew Meselson and Franklin Stahl. Matthew Meselson and Franklin Stahl. Meselson and Stahl performed the following experiment in 1958. They grew E. coli in a medium containing NH4Cl N15. N15 is the heavy isotope of nitrogen as the only nitrogen source for many generations. The result was that N15 was incorporated into newly synthesized DNA. N15 was incorporated into newly synthesized DNA as well as other nitrogen containing compounds. This heavy DNA molecule could be distinguished from normal DNA by centrifugation in cesium chloride CsCl density gradient. Cesium chloride density gradient. Please note that N15 is not a radioactive isotope and it can be separated from N14 only based on densities. They transferred the cell into a medium with normal NH4Cl N14 and took sample at various definite time intervals. Then they transferred the cell into a medium with normal NH4Cl N14 and took samples at various definite time intervals as the cell multiplied and extracted the DNA that remained as a double-stranded helix. The various samples were separated independently on cesium chloride gradient to measure the densities of DNA. Can you recall what centrifugal force is and think why a molecule with higher mass density would sediment faster? N15 DNA 20 minutes. Gravitational force N15 heavy. Hybrid N14 N15. N15 DNA N14 DNA. Hybrid. Separated by DNA. Separation of DNA by centrifugation. Centrifugation. Meselson and Stahl experiment. Thus, the DNA that was extracted from the culture one generation after the transfer from N15 to N14. N14 medium, that is 20 minutes. E. coli divides in 20 minutes. Had a hybrid of intermediate densities. DNA extracted from the culture. DNA extracted from the culture after another generation, that is after 40 minutes. Second generation was composed of equal amounts of the hybrid DNA extracted from the culture after another generation, that is after 40 minutes. Second generation was composed of equal amount of this hybrid DNA and the light DNA. 40 minutes light DNA. If E. coli was allowed to grow for 80 minutes, then what would be the properties of what would be the properties now? What would be the proportions of light and hybrid densities DNA molecule? Okay, it's 40, so 60 minutes. Is that 80? Yes, it is 80. 60. Minutes. 1, 2, 3, 4, 5, 6, 7, 8, 9, 10. 10 and 14 light DNA. 2 and 15, which will be already hybrid, and 15 and 14 hybrid DNA. Vector, a very similar experiment involving use of radioactive thymidine to detect distribution of newly synthesized DNA. Very similar experiment involving use of radioactive thymidine to detect distribution of newly synthesized DNA in the chromosome was performed on Vicia faba bean by Taylor and colleague. In 1958, the experiment proved that DNA in chromosome also replicates semi-conservatively. Vicia faba bean, Taylor and colleague. Observed contain one strand of parent DNA. Okay, semi-conservative DNA replication. Semi-conservative DNA replication observed contain one strand of parent DNA and other strand of newly polymerized synthesized DNA. This mode of replication is called semi-conservative mode of replication. Was observed by Meselson and Stahl in E. coli and later Taylor in fava beans, Vicia faba. Meselson and Stahl used cesium chloride gradient technique. Taylor and colleague used radioisotope thymidine. Radioisotope thymidine. रेडियोएक्टिव ओके को रेडियो आइसोटोप को रेडियोएक्टिव यहाँ पर मैंने रेडियो आइसोटोप लिख दिया रेडियोएक्टिव थाइमिडिन radioactive thymidine technique radioactive thymidine Taylor Vicia fava Taylor. Okay, Matthew Meselson and Franklin Stahl. NH4Cl. Heavy isotopes. N15 is heavy isotope of nitrogen. Centrifugation in cesium chloride density gradient. Cesium chloride density gradient. Excuse me. Okay, cesium chloride density gradient technique by Meselson and Stahl. E. coli is initially cultured, grown in N15 isotopes medium. Result into incorporation of N15 in DNA. Contain NH4Cl. A nitrogenous base is pair of DNA. क्या लिखा है? Contain NH4Cl as nitrogenous base pair. NH4Cl as nitrogenous base pair of DNA. Contain NH4Cl as nitrogenous base pair of DNA. E. coli subsequently cultured in an N15 isotope medium. Second में तो N14 में करते हैं. हाँ. E. coli subsequently cultured in N14 medium. E. coli subsequently cultured in N14 isotope medium. NH4, N14 chloride, NH4Cl, N14. E. coli progeny reproduction had been observed. Replication had been observed at a specific time interval, 20 minutes to 40 minutes, and then finally 60 minutes. DNA. N15 NH4Cl after 20 minutes. This is hybrid. 20 and then finally 40 after 40 minutes. 2 light DNA, two hybrid, after 60 minutes light, this is 40, after 60 minutes light form will increase. Keeping mixed N14 to N15 static. Keeping mixed N14 to N15 static. Okay. What could be the percentage possibility of lighter band on N14 DNA after 60 minutes? In 80 minutes. Okay, तो questions है. N15 DNA. N15 DNA. N15 DNA to 20 minutes. hybrid N15, N14. N15 DNA. Generation 2. Separation by cesium chloride density gradient. Cesium chloride density gradient. Gravitational force. Astaghfirullah. Hick. And 15. And 15. And 15. Heavy. Hybrid and 14 and 15 and 14 and 15. Light. Hybrid. Transcription. Molecular basis of inheritance. Nucleic acid, molecular unit of inheritance. Nuclein, first observed by Friedrich Mischer. Nuclein, first observed by Friedrich Mischer. DNA, deoxyribose. Absence of oxygen at second carbon position of sugar has thymine in place of uracil. Deoxyribose. Deoxyribose sugar has no oxygen at second carbon. Absence of oxygen at second carbon position of sugar has thymine in place of uracil. DNA. Nitrogenous base, purine and pyrimidenes. Purine, 9-membered double ring structure and present. Purine, 9-membered double ring structure present. Purine, 9-membered double ring structure and present. Position of N at in purine, 1379, 1379, 1379. Position of N. Purine, adenine and guanine, adenine and guanine. Position of N at 1379. Pyrimidine, 6-membered single ring structure. Position 6-membered. They are 6-membered single ring structure. Position of N at 1 and 3. 1 and 3. Uracil, thymine and cytosine. Uracil, thymine and cytosine. Pyrimidine. Purine, adenosine, adenine, NH2 present on 6th carbon. Adenine ma NH2 6th carbon ma present हुन्छ. Adenine ma NH2 6th carbon ma present हुन्छ. Guanine ma NH2 second carbon ma र double carbonyl groups C double bond O 6th carbon ma हुन्छ. NH2 present on second carbon and carbonyl group present on 6th carbon. This is for guanine. Okay. Pyrimidine. Cytosine, NH2 4th carbon ma हुन्छ. Carbonyl group second carbon ma. NH2 4th carbon ma. Okay, second carbon 4th ma थियो. Double bond O second carbon ma. Carbonyl group 4th position ma NH2. Thymine, CH3 group is present on 5th methyl uracil. 5th methyl uracil. Uracil double bond O. Carbonyl group is present on 2nd and 4th carbon in ring. 2nd and 4th carbon in ring. Okay. Adenine, guanine, cytosine and uracil. Adenine, NH2 at 6th position of carbon. Guanine, NH2 at carbonyl group. Carbonyl group at 6th position of carbon. NH2 at 2nd carbon. Cytosine, carbonyl group at 2nd carbon and NH2 at 4th carbon. Uracil, C double bond O, carbonyl in both 1 and 2 and 4. Thymine, THYMine. Thymine is uracil but with methyl CH3 in 5th position carbon. 6th carbon, NH2 6th carbon. Guanine, carbonyl group present on 6th carbon. NH2 present on 2nd carbon. Cytosine, C double bond O. Where is it? Second and 4th. Pentose sugar, ribose sugar, RNA, oxygen at 2nd carbon and deoxyribose sugar, absence of oxygen at 2nd carbon. Okay. CH2OH. OH, OH, OH. This is ribose. Ribose. In ribose sugar, in DNA deoxyribose CH2 OH, OH. Here we have H, deoxyribose H. Okay. Phosphate PO4 3 minus. Actually, it is H3PO4. That was with P minus 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 P double bond O. This is phosphate group. Okay. 3 prime ma OH free end हुन्छ. 5 prime ma PO4 3 minus. Sugar. Base. Nitrogenous base. This is N-glycosidic bond. N-glycosidic bond. This one is N-glycosidic bond, and this one is phosphoester bond. Phosphoester bond. Phosphoester bond. Phosphate group. Phosphoester bond. एक phosphate दूसरा सुगर. Phosphoester bond. This one is again phosphoester bond. 2 phosphoester bond is equals to 1 phosphodiester bond. Glycosidic bond. And glycosidic bond, phosphate bond, phosphoester bond. Okay. And glycosidic bond, purines and pyrimidines. I think मैंने ऑफ कर दिया ना. लो ऑन कर देती हूँ, खुद के लिए हाइड कर देती हूँ. रुको. कहाँ से हाइड करते हैं? यहाँ पर था तो एक कुछ हाइड करने के लिए. नन. Purine 9 member double. Okay, ये हो गया था. I have to complete from this point. And glycosidic bond. Purines. Glycosidic bond. Both for purines and pyrimidines. Purines and purines C191 N9 N9. C1 of sugar, N9 of nitrogenous base, whereas pyrimidines C1 of pentose sugar and N1 of pyrimidine. N of nitrogenous base. Okay, N1 C1 9 N9 C1 N9 for purine. C1 N9 pyrimidine and purines respectively. Okay. Bases: adenine, guanine, cytosine, thymine, uracil. RNA: adenosine, guanosine, cytidine, uridine. Nucleoside में ना nucleotide होता है adenosine monophosphate, guanylic acid, cytidyllic acid, uridylic acid. In DNA, deoxyadenosine, deoxyguanosine, deoxycytidine, deoxythymidine, deoxythymidine, deoxycytidine, deoxyguanosine, adenosine, guanosine, cytidine and thymidine. Deoxyadenosine monophosphate, deoxyguanylic acid, deoxycytidyllic acid and deoxythymidyllic acid. Formula: N-1 is equals to number of phosphodiester bond. N equals to glycosidic bond. Where N is the number of base pair. Nitrogenous bases present in a polynucleotide chain. N-1. If base equals to N, N-1. If base is equals to N, N-1. Okay. If base equals to N. If a single polynucleotide chain has 4 bases. इसको ना कहीं लिखना पड़ेगा शायद. If a single polynucleotide chain chain 4 bases are observed. In a single polynucleotide chain 4 bases are observed. What would be the total number of phosphodiester bond and glycosidic bond? Glycosidic bond is equals to number of bases. Phosphodiester bond is equals to N-1. Phosphodiester bond is equals to N-1. Number of phosphate bond N-1. If bases N. Number of bases -1. That is phosphodiester bond. 2 phosphoester bond is equals to 1 phosphodiester bond between two nucleotide. Pentose sugar 3 prime sugar 3 क्या बोल रही है concentrate. Pentose sugar 3 prime carbon free hydroxyl group 3 prime carbon free hydroxyl group OH group 3 prime carbon of sugar free hydroxyl group OH 5 prime carbon free phosphate group PO4 3 minus group. Okay. Attack on 3 prime carbon PO4 of 5 prime carbon OH hydroxyl group. It can let PO4 3 minus attack to it. Okay. DNA is a polynucleotide complementary form of anti-parallel strand polynucleotide 5 prime to 3 prime 3 prime to 5 prime. Complementary between adenine and thymine, guanine and cytosine. DNA between adenine and thymine 2 hydrogen bond, between guanine and cytosine 3 hydrogen bond. For DNA adenine and thymine 2 hydrogen bond, between guanine and cytosine 3 hydrogen bond. 3 hydrogen bond and 2 hydrogen bond each. How many glycosidic bonds are present in DNA when the DNA contains 6 base pair? Glycosidic N equals to 6, glycosidic bond be 6, 6 glycosidic bond. How many glycosidic bonds are present in DNA when DNA contains 6 base pair? Oh, base pair बोला है, not base. 6 into 2, 12. Hmm. 6 into 2, 12. Silent features of DNA. Complementarity, base pairing. Anti-parallel feature, right-handed coiled, right-handed coiled, dexterous rotation. Watson and Crick model 1953. Watson and Crick model 1953. BDNA. In one turn, there are 10 base pair in one turn. Pitch of helix, 10 base pair. Pitch of helix, 10 base pair. One turn, 10 base pair. Okay. Pitch of helix, 3.4 nanometer or 34 Armstrong. 3.4 nanometer or 34 Armstrong. Distance between two bases, 0.34 nanometer or 3.4 Armstrong. 1 nanometer is equals to 10 to the power minus 9 meter. 1 nanometer is equals to 10 to the power minus 9 meter. 3.4 Armstrong. 3.4 nanometer, 34 Armstrong. Pitch of helix, okay? Number of base pair in somatic diploid cell of human, 6.6 into 10 to the power 9 base pair. 6.6 into 10 to the power 9 base pair. 6.6 into 10 to the power 9 base pair for diploid somatic cell of human. Length of DNA 2.2 meter for human. 2.2 meter for human. 2.2 meter for human. 6.6 into 10 to the power 9 base pair 2n. 3.3 into 10 to the power 9 base pair n. Okay, 3.3 into 10 to the power 9 base pair n. Base pair of E. coli is equals to 4.6 into 10 to the power 6. Base pair of E. coli is equals to 4.6 into 10 to the power 6 base pair. Length of DNA 1.36 meter. Base of length of DNA of E. coli 1.36 meter. Diploid base pair of E. coli diploid 4.6 into 10 to the power 6. 4.6 into 10 to the power 6. meter. 4.6 into 10 to the power 6 base pair. Base pair of E. coli is equals to 4.6 into 10 to the power 6 base pair of E. coli is equals to 4.6 into 10 to the power 6 base pair. 4.6 into 10 to the power 6 base pair. Stacking features, main framework, sugar phosphate structure, inside branch bases. In BDNA, bases are in one plane stacking over one another. Main framework, sugar phosphate structure, inside branch bases. In BDNA, bases are in one plane stacking over one another, each other in a polynucleotide chain. Stacking feature, Allah knows better. Stacking feature, main framework, sugar phosphate structure, sugar and then phosphate. Inside branch bases. Sugar phosphate bond, okay, bases. made in sugar phosphate, main backbone sugar phosphate. Bases are projected inside. Okay, in bDNA, bases are in one pair stacking over one another each other in a polynucleotide chain. Chargaff rule proposed by Chargaff based on complementarity base pairing. Postulate: sum of purine is equals to sum of pyrimidine. Sum of purine is equals to sum of pyrimidine. P, that is A plus G, is equals to C plus T. A plus G is equals to T plus C. The ratio A plus G upon C plus T is equals to 1. A plus G upon C plus T या फिर T plus C is equals to 1. T और ये C, TC, T plus C is equals to 1, constant for all species if the ratio of A plus T, G plus C. The ratio of A plus, क्या लिखा है मैंने, मुझे कुछ सही आता है. A plus T, G plus C. Specific value for different species. Prokaryote less than 1, A plus T, G plus C less than 1. For eukaryote, A plus T, G plus C greater than 1. DNA genetic material. Okay. Chargaff rule. Now sum of pyrimidine is equals to sum of purine. Ag is equals to T plus C. Chargaff ले दिया. A plus T upon G plus C is equals to 1, constant for all species. फिर से गलत. A plus G upon T plus C is equals to 1. A plus G is equals to T plus C. Sum of purine is equals to sum of pyrimidine. This is 1. Number 2, A plus G. Ratio of purines upon pyrimidine, T plus C, is equals to 1, constant for all species. But 1 A plus T upon G plus C should be greater than 1 for eukaryotes, and A plus T upon G plus C will be less than 1 for prokaryotes. Search for DNA as genetic material. 3 different experiment. Okay, search for DNA as genetic material. 3 different experiment. Griffith the transforming principle. Biochemical principle. Biochemical nature of transforming principle. Biochemical nature of transforming principle and Hershey-Chase experiment. Hershey-Chase experiment. Radioisotopes P32 and S35. Griffith transforming principle. Biochemical nature of transforming principle. Hershey-Chase experiment. Radioisotopes P32 and S35. Unequivocal proof of DNA as genetic material. Unequivocal proof of DNA as genetic material. Griffith experiment 1928. Test organism Streptococcus pneumonia bacteria. Streptococcus pneumonia, 2 strains, one rough strain, other one is S strain, smooth strain, that is virulent. Absence of mucus layer, non-virulent, R strain. Presence of smooth layer due to polysaccharide, virulent, harmful, S strain. Experiment of Griffith, R live strain plus mice. Mice lives as a strain, heat kill plus mice. Mice lives as a strain, live plus mice. Mice dies. R strain live plus as a strain, heat killed plus mice. Mice dies. Conclusion: R strain bacteria is transformed because of the virulent gene of S strain. R strain bacteria is transformed because of the virulent gene of S strain, and Griffith referred this transformation as transforming principle. Instead transforming principle and Griffith लिखे. Streptococcus pneumonia bacteria कहती हो. Streptococcus pneumonia 1928. Biochemical nature of transforming principle. 3 साइंटिस्ट पढ़े थे. Avery, McCarthy and MacLeod. Avery, MacLeod and McCarthy. Biochemical nature of transforming principle experiment. Isolation of three enzymes: RNAs, peptidase, and DNAs. Protease okay, peptidase. Protease, RNAs, and DNAs. Denaturation of protein by protease, denaturation of RNA by RNAs, and denaturation of DNA by DNAs. Same set of Griffith experiment was used. R strain live plus S strain heat killed plus protease. Transformation observed: R strain live, S strain heat killed plus RNAs. Transformation observed in both cases. No role of protein in transformation. No role of protein along with RNA in transformation. R strain plus S strain heat kill plus DNAs. Transformation. No transformation, जिसमें तो transformation was visible होना चाहिए था. नहीं सही है. No transformation was visible करने की DNA लाइनें distort कर दिया. Transformation observed in both the cases. No transformation. DNA was the biochemical nature of transformation. DNA was the biochemical nature of transformation, but still could not consider DNA as genetic material. Hershey-Chase Experiment. Unequivocal observation of DNA as genetic material by using radioisotope method. P32 as a component of DNA, S35 as a component of protein. P32 as a component of DNA, S35 as a component of protein. P32 as a component of DNA, S35 as a component of protein. Included bacteriophage, virus that infect bacteria E. coli. Bacteriophage and bacteria E. coli. 3 steps: infection, binding, and centrifugation. Infection, blending, not binding. Infection, blending, and centrifugation. Infection, blending, and centrifugation. Infection, blending, and centrifugation. Centrifugation. DNA P32, protein S35, bacteriophage. DNA P32, protein S35. Radioactive DNA versus radioactive protein. Infection. Infect the E. coli. No radioisotope found inside cell. Protein is outside. Here, radioactive DNA P32. Step blending to separate the two entities. Okay, blending. Protein remains outside the cell. Protein code remains outside the cell. Genetic material is inside the cell. E. coli. Third step, centrifugation. Radioisotopes P32. Bacterial mass palette. Radioisotopes P32. Supernatant. S35 was found in supernatant. S35. Radioactive isotopes on supernatant. S35 protein. Okay, bacterial mass palette. Not radioactive isotope. P32. Bacterial palette by P32. Genetic material of virus was detected in bacterial palette or bacterial mass. P32 was genetic material of virus. P32 was genetic material of virus was passed to bacteria as the palette of bacteria contain radioisotopes, part of DNA. P32 was genetic material of virus was passed to bacteria as the palette of bacteria contain radioisotopes, part of DNA. DNA is genetic material. Packaging of DNA in chromosomes are central dogma. उसके बाद process of replication for replication. Packaging of DNA in chromosome. Length of human DNA 2.2 meter. Chromosome. Condensation and packaging of DNA in small length. Carrier functional gene. Carrier functional gene. Condensation and packaging of DNA in small length. Okay. Chromosome. Length of human DNA 2.2 meter. Condensation and packaging include histone protein. Histone protein चार्टा टाइप कौन सा है? Basic protein due to lysine and arginine. Basic protein due to lysine and arginine consists 5 histone unit. H1, H2A, H2B, H3 and H4. H1, H2A, H2B, H3 and H4 histone consist of 5 histone unit. Okay. Basic protein lysine and arginine consist of 5 histone protein. H2A, H2B and H3 and H4 are okay. These 4 are present in duplet form. Okay, they are present in duplet form and they form octamer complex. H2A, H2B, H3 and H4 are found in duplet form. Duplet form. Duplet form of histone. Octamer complex. Octamer complex. Octamer complex. Octamer complex. Nucleosome. Nucleosome 200 base pair of DNA. Acidic DNA 3/4 turn. Bison string converted to solenoid appearance. Octamer H1 histone. DNA is condensed as chromatin fiber. DNA is condensed as chromatin fiber. (200 base pair of DNA. Acidic DNA. Bison string converts to solenoid appearance. DNA is condensed as chromatin fiber. H1 histone. Octamer. Nucleosome. Linker DNA small fragment. (200 base pair of DNA. Acidic DNA. 3/4 turn. Bison string. Basic protein lysine. DNA acidic. Okay, lysine and arginine. Nucleosome. Linker DNA small fragment. Bison string in chromatin structure. Euchromatin. They are lightly stained. Active transcription. Transcriptionally active chromatin. Loosely packed, lightly stained. Okay, those are euchromatin. Okay, euchromatin. Active transcription. Euchromatin active transcription. Loosely packed, lightly stained. Loosely packed, lightly stained. Loosely packed and lightly stained. Transcriptionally active chromatin. Transcriptionally active chromatin. Loosely packed, lightly stained. Transcriptionally active chromatin. Then we have heterochromatin. Heterochromatin. Densely packed, stains dark, transcriptionally inactive. Heterochromatin. Densely packed, stains dark, transcriptionally inactive chromosome. NHC. Non-histone protein. Non-histone chromosomal protein. Non-histone chromosomal protein helps in packaging in higher organism. Role in condensation of chromatin. Structure and regulatory role in condensation of chromatin as well as structural and regulatory role. Structural and regulatory role. Structural and regulatory role. Structural and regulatory role. histone chromosomal protein. Okay, euchromatin, transcriptionally active, loosely packed, lightly stained chromatin. Heterous chromatin are densely packed, darkly stained, and transcriptionally inactive. By the way, normal. Central Dogma by Francis Crick. Central Dogma by Francis Crick. It includes the principle of conversion of gene sequence, base pairing sequence into gene expression. It includes the principle. It includes the principle. It includes the principle of conversion of gene sequence, base pairing sequence into gene expression. Central dogma include two important process steps: transcription and translation. Central dogma by Francis Crick. Central dogma by Francis Crick. Central dogma by Francis Crick. It includes the principle of conversion of gene sequence, base pair sequence. It includes the principle of conversion of gene sequence, base pair sequence into gene expression. Conversion of gene sequence into gene expression. Gene sequence into gene expression. It includes the principle of conversion of gene sequence into gene expression. Gene sequence, base pair sequence into gene expression. After almost a year, I don't go there often. It includes the principle of conversion of gene sequence, base pair sequence into gene expression. Central dogma includes two important process steps: transcription and translation. It includes the principle of conversion of gene sequence, base pair sequence into gene expression. Central dogma includes two important process steps: transcription and translation. What's the quirrell? Transcription, translation. DNA replication, transcription, mRNA, translation, protein. DNA replication. Okay, central dogma. Francis Crick. HIV retrovirus does not follow central dogma. Central dogma not followed by retrovirus HIV. Francis Crick. Okay. Reverse transcription or terminism. Transcription or terminism by some viruses, restricted only to certain virus like retrovirus. RNA becomes DNA. RNA becomes DNA. DNA replication, transcription, mRNA, translation, protein. Replication of DNA molecule. DNA dependent DNA polymerase. Okay. Other than this, we have helicase for unwinding and SSB protein, single stranded binding protein, to avoid the base pairing between the two strands. Then we have another enzyme that is known as primase, because DNA can't synthesize its own new strand. They need RNA primer. Primase helps in formation of primer. Then few more enzymes are there: SSB, helicase, primase, DNA ligase, deoxyribose, DNA polymerase है ना. Then DNA ligase. DNA polymerase, helicase, primase, ligase. Okay, topoisomerase is also there. Topoisomerase and ligase. Topoisomerase and ligase. Topoisomerase helps in preventing positive supercoiling. DNA gyrase in prokaryotes. Topoisomerase को ही क्या बोलते हैं? DNA gyrase. DNA gyrase in prokaryote. DNA gyrase in prokaryotes helps in binding or attaching short fragment of DNA. Helps in binding or attaching short fragments of DNA. Short fragments of DNA. Duplication of DNA molecule is known as DNA replication. Duplication of DNA molecule. Duplication of DNA molecule. DNA dependent DNA polymerase included in replication. DNA dependent DNA polymerase needs a DNA template based on the principle of complementarity. Based on the principle of complementarity. Polymerization of new DNA strand is observed. DNA dependent DNA polymerase needs a DNA template based on the principle of complementarity. In prokaryote replication. Okay, in prokaryote replication is observed at once. Single point ORI, origin of replication. Single starting point or origin of replication. Single start point or origin of replication. It starts replicating at one end of DNA and continuously fulfill the duplication until the end of DNA is observed. Reason: DNA is single and circular. Single circular. In prokaryotes replication. In prokaryote, replication is observed at once. Single starting point or origin of replication ORI. Single start point or origin of replication. It starts replicating at one end of DNA and continuously fulfills the replication until the end of the DNA is observed. Reason: DNA is single and circular. It starts replicating at one end of DNA and continuously fulfills the replication until the end of DNA is observed. In eukaryote replication, there are multiple origin of replication. In eukaryote, ORI is observed at many sites. Okay. Process of replication. Besides the DNA polymerase, many other enzymes are involved. DNA polymerase, helicase, primase, ligase, and topoisomerase. Besides DNA polymerase, many other enzymes are there. DNA polymerase, helicase, primase, ligase, and topoisomerase. Helicase, primase, ligase, and topoisomerase, along with single-stranded binding protein. Okay. In prokaryotes, mainly three types of DNA polymerase are found: DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. Three types of DNA polymerase in prokaryotes. Okay. DNA polymerase. All the DNA polymerase do polymerization in 5' to 3' direction. DNA polymerase do polymerization in 5' to 3' direction. Highest rate of polymerization is observed in DNA polymerase 1. Not given. In DNA polymerase, 200 base pair per second. 2000 base pair per second. Highest rate of polymerization is observed in DNA polymerase, 2000 base pair per second. DNA polymerase also helps in proofreading for correction and accuracy. DNA polymerase also helps in proofreading for correction and accuracy. Fourth point, DNA polymerase can't do de novo new synthesis. It always requires a short fragment of RNA primer, RNA to polymerize the new DNA. All the DNA polymerase do polymerization in 5' to 3' direction. Polymerization हुन्छ 5' to 3' direction. Highest rate of polymerization 2000 base pair per second. 2000 base pair per second. DNA polymerase also helps in proofreading for correction. Proofreading helps in proofreading for correction and accuracy. Proofreading Exonuclease activity is observed generally in 3' to 5' direction. 3' to 5' direction exonuclease activity, but 5' to 3' exonuclease is observed by DNA polymerase 1. DNA polymerase 1 को कारणले गर्दाखेरि 5' to 3'. DNA polymerase 1 is used to disconnect the primer. DNA polymerase 1 is used to disconnect the primer. Primer disconnection. DNA polymerase 1. DNA polymerase 1. या अल्लाह मैं प्लीज कर सकूं. DNA polymerase also helps in proofreading for correction and accuracy. Proofreading exonuclease generally works in 3' to 5' direction. Exonuclease activity is observed generally in 3' to 5' direction, 3' to 5' direction, but DNA due to the presence of DNA polymerase 1, it works in 3' to 5' to 3' direction. Absorbed by DNA, but 5' to 3' exonuclease is absorbed by DNA polymerase 1. Okay, DNA polymerase 1 is used to disconnect the primer. DNA polymerase can't do de novo synthesis. It always requires a short fragment of RNA primer to polymerize the new DNA. 2000 base pair per second. 2000 base pair per second. Proofreading, correction, and accuracy. Helicase helps in unwinding of DNA that result in replication fork. Helps in unwinding of DNA that result in replication fork. SSB, single-stranded binding protein, helps in maintenance of single strand for replication. Helps in maintenance of single strand. Helps in maintenance of single stranded for replication. Helps in maintenance of single strand for replication. Helps in maintenance of single strand for replication. It prevents complementarity after unwinding. Single stranded. We'll take a break of 10 minutes, okay? First slot is done. Single-stranded binding protein helps in maintenance of single strand for replication. Single-stranded binding protein. Single-stranded binding protein helps in maintenance of single strand for replication. Helps in maintenance of single strand for replication. It prevents complementarity after unwinding. Helps in maintaining of single strand for replication. Helps in maintenance of single strand. Helps in maintenance of single strand for replication. It prevents complementarity after unwinding. Topoisomerase, also known as DNA gyrase in prokaryotes. DNA gyrase. It relieves the strain in DNA. It relieves the strain in DNA. Helps in preventing positive supercoiling. Preventing positive supercoiling. DNA gyrase in prokaryotes. Primase helps in formation of RNA primer. DNA ligase helps in binding or attaching short fragment of DNA. Helps in binding and attaching short fragments of DNA. DNA primase. DNA ligase helps in binding or attaching short fragment of DNA. Replication fork. Replication. Both the strands are involved in replications. 3' to 5'. 5' token polymerase needs primer. From 5' to 3' polymerization or replication. 5' to 3' polymerization or replication. RNA primer is removed with the help of DNA polymerase 1. RNA primer is removed with the help of DNA primase 1. RNA primer is removed with the help of DNA polymerase 1. Polymerase needs primer. 5' to 3' polymerize. जाओ सारो वक्कै लाग्दो. Needs. लास्ट ईयर की बात थी. मुझसे नहीं होगा. Both the strands are involved in replication. Needs primer. 5' to 3' polymerization or replication. Allah, I need your help, please. Helicase, single-stranded binding protein and topoisomerase. Helicase, single-stranded binding proteins and topoisomerase. Supercoiling. Prevent positive supercoiling. DNA gyrase. Enzymes. Ligase SSPN2 polymerase unwinding of DNA and making a single strand. Connecting small fragments of DNA helps in binding or attaching short fragments of DNA. Primase. DNA polymerase 1. Leading strand, discontinuous or lagging strand. Okazaki fragments are present. Many short RNA primer fragments are observed. DNA ligase joins those small fragments. Polymerization. Treatment. Five prime to 3 prime. 3 prime. DNA polymerase requires 3 prime OH. DNA provided by RNA primase. Free end for polymerization. Free end for polymerization. कहाँ लिखा है ऐसा? वहाँ पर DNA polymerase requires 3 prime OH provided by RNA prime. Okay, DNA polymerase requires 3 prime OH provided by RNA primer. Free end for polymerization. Free end for polymerization. DNA polymerase requires 3 prime OH. 3 prime OH. Provided by RNA polymerase. Provided by RNA polymerase. Provided by RNA polymerase. Free end for polymerization. Semi-conservative DNA replication contain one strand of parent DNA and other strands of newly polymerized synthase. Okay, one strands of parent DNA and other strand of newly polymerized synthesized. Observed by Meselson and Stahl, Matthew Meselson and Franklin Stahl in E. coli. Khanera E. coli in E. coli. Okay. Later by Taylor and colleague in Vicia fava beans, 1958. Meselson Stahl cesium chloride gradient technique. Taylor used active thymidine technique. Radioactive thymidine technique. Radioactive thymidine technique. अब ट्रांसक्रिप्शन पढ़ेंगे. 10 मिनट का ब्रेक लेंगे. ओके हिमावरी, हम लोगों का स्लॉट 1 कंप्लीट हो गया है ना, इसलिए 10 मिनट का ब्रेक लेंगे. ओके 7 मिनट पहले ही कंप्लीट हो गया था. चलो टी स्लॉट 1 कंप्लीटेड. ओके मुझे यहाँ पर आना होगा 4:10 पर आना होगा. I don't have another. मैं 10 मिनट का ब्रेक लेके गई थी लेकिन पहले ही आ गई. ओके, स्टार्ट करते हैं. Slot 2 begins. इसको भी ऑफ कर देती हूँ अभी. Like replication, transcription is also based on complementarity. Transcription. Okay. In replication, both the strands are template. Strand, that is, replication observed on both strands. Like replication, transcription is also based on complementarity. In replication, both the strands are template strand, that is replication observed on both strand. But in transcription, only one strand, template strand. Okay, like replication, in replication, both the strands are template strand, that is replication observed on both strand. In replication, both the strands are template strand, that is replication observed on both strand. Replication observed on both strand. But in transcription, only one strand, template strand. If both the strands of DNA is used as a template for conversion into mRNA strand, GAG. ATC GAMRNA. Okay, this is RNA. Both strands of mRNA codes for different amino acid polypeptide chain protein. The situation is not possible as its chains alter gene expression. Both the mRNA can be complementary to each other, making the complementary strand. Gene expression is not observed. Okay, transcriptional unit. Choro. Like replication, transcription is also based on complementarity. Like replication, transcription is also based on complementary. Like replication, transcription is also based on complementarity. Base pairing. Complementarity, base pairing. Like replication, transcription is also based on complementarity, base pairing. Transcription, okay. In replication, both the strands are template strand. In replication, both of the strands are template strand. In replication, both of the strands are template strands. In replication, both the strands are template strand. Replication observed on both sides. Replication observed. Both sides. Replication, transcription also known as complementary base pairing. In replication, both the strands are template strand, that is replication observed on both strands. But in transcription, only one strand, template strand. Replication is seen only in one strand. Transcription, okay, go. Replication. Transcription. Transcription is seen only in one strand. STRAND strand. Transcription is seen only in one strand. Template. TEMP. Template strand. Reason behind one template strand. Reason behind one template strand. Reason, alhamdulillah. If both the strands of DNA is used as template for conversion into mRNA, both strands of mRNA codes for different amino acid. Strand of DNA used as template for conversion into RNA strand. Both strand of mRNA codes for different amino acid. If both strand of DNA is used as a template for conversion into mRNA strand, both mRNA strand codes for will code for different amino acid. Polypeptide chain. Protein. The situation is not possible as it change altered gene expression. If both the strands of DNA is used as template for conversion into mRNA strand, both mRNA strand will code for different amino acid, polypeptide chain, protein. This situation is not possible. This is possible as it changes and alters gene expression. GAGCTA. This is a one strand, the other strand is CT. यहाँ पर ए होता है ना, ए टी को जगह में ए C को जगह पे U था, C को जगह में G, T को जगह में U, C को जगह में G, G को जगह में C, A को जगह में U, T को जगह में A, G U G, C U C, G A U, G U G, C U A. This is a mRNA strand. Okay. Okay, amino acid 1, amino acid 2. Okay, amino acid 1, amino acid 2, amino acid 3, and amino acid 4. Amino acid 4. mRNA. mRNA, therefore A equals to U and T equals to A. Okay. Reason behind one template strand. If both the strand of DNA is used as template for conversion into mRNA strand, both mRNA strand will code for different amino acid polypeptide chain protein. This is not possible as it changes and alters gene expression. As it changes and alters gene expression, GAG, CTA. GUG, CUA. This is amino acid. Okay, 3 set, 1 amino acid. Okay. DNA. Both strands of mRNA codes for different amino acid polypeptide chain protein. The situation is not possible as it changes, alters gene expression. CUCGAU mRNA. GAG CUA. Both the mRNA, okay. CGUA, CGGC, AUUA. Both the mRNA can be complementary with each other. Both the mRNA can be complementing each other, making complementary strands. Gene expression is not observed. Okay, these are the two reasons. Both mRNA and messenger RNA can be complementing. Each other. Complementing each other, making complementary strand. Making complementary strands. STRA and DS strands. Both mRNA can be complementing each other, making complementary strands. In this case, gene expression is not observed. Gene expression, EXPRASSIN, expression is not observed. Gene expression is not observed. CUCGAU. CUCGAU, mRNA. GAG CUA. This is C, okay. Both mRNA can be complementing each other, making complementary strand. Excuse me. and be complementing each other, making complementary strands. Gene expression is not observed. Transcription unit. For the process of transcription, the segment of DNA is specified and certain reasons are acknowledged, identified. Includes three side components. Transcription unit. For the process of transcription, the segment of DNA is specified and certain reasons are acknowledged or identified. Transcription unit. Transcription unit. Transcription unit. Transcription unit. Process of transcription. For the process of transcription. For the process of transcription, the segment. For the process of transcription, the segment of DNA is specified. For the process of transcription, the segment of DNA is specified. Certain reasons are acknowledged or identified. The segment of DNA is specified. Certain reason or certain reason are acknowledged or identified. Includes three site components: promoter site reason, structural gene, and terminator site or reason. Promoter site reason includes sites or components. Promoter site reason represents the reason which initiates the transcription. Structural gene or reason. Structural gene flanked by promoter and terminal. Structural gene represents the segment of DNA which undergoes conversion. And finally, terminator site reason terminates the transcription. Structural gene, promoter site, structural gene, and terminator site. Promoter, transcription starts here. Terminator, structural gene, template strand and coding strand. Promoter site or reason, structural gene, and terminator site or reason include three site or components. Transcription unit. Transcription unit has three components. Includes three sites or components. Components of transcription unit include three sites or components: promoter site or reason, structural gene, terminator site or reason. Promoter site reason. Promoter reason, structural gene, structural, structural gene. Promoter site reason, structural gene, and terminator site or reason. Terminator site / reason. Promoter site reason, structural gene, and terminator site or region. Terminator site, promoter site. Promoter region represents the region which initiates the transcription. Sequence of base that is recognized by RNA polymerase is different in prokaryote and eukaryotes. At the upstream of promoter, the recognition sequence for prokaryotes and eukaryote transcription are present. Terminator site terminates the transcription. AAT base sequence for prokaryote recognition sequence in is क्या लिखा है मैंने for eukaryote it is Hogness box. Pribno box, Hogness box. Coding and non-coding sequence in eukaryotes. Structural gene, monocystronic. And मैंने ये वो नहीं किया है क्या, ऑफ नहीं किया है क्या chats? Promoter site. Promoter site represents the region which initiates the transcription. Promoter represents the region which initiates the transcription. Represents the region which initiates the represents the region which initiates the transcription. Represents the region which initiates the transcription. Promoter site or region represents the region which initiates the transcription. Sequence of bases that is recognized by RNA polymerase is different in prokaryotes and eukaryotes. SEQUENC. Sequence of bases recognized by RNA polymerase. Sequence of base recognized by RNA polymerase. Sequence of base recognized by RNA polymerase is different. Sequence of base recognized by RNA polymerase is different. In prokaryotes and eukaryotes. Sequence of bases that is recognized by RNA polymerase is different in prokaryotes and eukaryotes. Okay. Promoter site represents the region which initiates the transcription. mRNA बनिन्छ DNA देखि. Sequence of bases recognized by RNA polymerase is different in DNA dependent DNA polymerase in replication. Okay, RNA polymerase को कुरा गर्दा sequence of bases recognized by RNA polymerase is different in prokaryotes and eukaryotes. At the upstream At the upstream of promoter, the recognition sequence for prokaryotes and eukaryotes transcription are present at the upstream. At the upstream of promoter, the recognition sequence for prokaryotes, the recognition sequence the recognition sequence for prokaryotes recognition sequence for prokaryotes and eukaryotes the recognition sequence for prokaryotes and eukaryotes transcription are present. Eukaryotes transcription are present. At the upstream of promoter, the recognition sequence for prokaryotes and eukaryote transcription are present. At the upstream of promoter. The recognition sequence for prokaryote and eukaryote transcription are present. The recognition sequence for prokaryotes and eukaryote transcription are present. Structural gene TAT base sequence for prokaryotes. For eukaryotes, it is Hogness box. TAT base sequence. For prokaryotes, recognition sequences Pribno box. Pribno. Let me check once. Recognition sequence for prokaryotes TAT or Pribno, PRIB, NOW, Pribno box. TAT 35 sequence TTGACA and the 10 sequence TAT or Pribno box. Excuse me. Recognition sequence for eukaryotes. The specific DNA sequence that are recognized by protein recognition sequence of eukaryotes. Hogness box. Hogness. Core promoter region of genes in Hogness box and Pribno box. Pribno's box is the bacterial homologue of the Tata box that has a shorter sequence. Okay. TATA base sequence for Pribno box. For Pribnos TATA, okay, Pribno box. Hogness box Tata. represents the region which initiates the transcription promoter site. Okay, sequence of bases recognized by RNA polymerase is different in prokaryotes and eukaryotes. Prokaryotes र eukaryotes मा base sequence different हुन्छ. जसलाई कसले recognize गर्छ RNA polymerase ले. Okay, at the upstream of promoter, the recognition stream for prokaryotes and eukaryotes transcription are present. Upstream of the promoter, the recognition sequence for prokaryotes and eukaryote transcription are present. At the upstream of promoter, the recognition sequence for prokaryotes and eukaryotes are present. Eukaryotes transcription are present. For prokaryotes, prokaryotes, it's pribno. For prokaryotes, it's PRIB, prib now box. Sequence TATAAT, pribno box. For eukaryotes, 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 it's Hogness box. Tata, TATA, Tata. This is base sequence. Okay, Tata base sequence. For prokaryotes, okay, Tata at base sequence. For prokaryotes, recognition sequence, pribno box. Base sequence, recognition sequence, SEQUENC, recognition sequence, base sequence. Okay, TATAT, base sequence for prokaryotes. Recognition base sequence. Recognition sequence, you base sequence. Okay, recognition sequence is pribno box, Hogness box. Coding and non-coding sequence in eukaryotes. Structural gene monocistronic. Monocistronic only form one type of polypeptide. Monocistronic only form one type of polypeptide. Cistron, functional gene unit. Cistron, SISTRONE. Cistron is functional gene sequence. In prokaryote, structural gene polycistronic. Okay, structural gene polycistronic forms more than one type of polypeptide. Forms only one polypeptide भनेपछि मोनोसिस्ट्रोनिक भनेको एउटा मात्रै polypeptide बनाउँछ. प्रोकार्योट्समा polycistronic हुन्छ, त्यसै कारण एउटा भन्दा धेरै polypeptide फर्म गर्छ. ओके, सिस्ट्रोन के हो? फंक्शनल युनिट हो युकार्योट्सको लागि. के सिस्ट्रोन functional unit of functional gene sequence. Monocistronic consists of two types of segment. I think मेरा नेट चल रहा है. Monocistronic consists of two types of segment. Monocistronic consists of two types of segment in structural gene. Eukaryotic. Exon, intron. Exon express cistron. Segment which express coding. Intron and cistron. Intervening, intron. Intervening, cistron. Segment which does not express or interfere. Coding and non-coding sequence. Coding and non-coding sequence. Okay, coding. In eukaryotes, eukaryotes, in eukaryotes, structural gene monocystronic. structu, structural gene monocystronic. Coding and non-coding sequence in eukaryotes, structural gene monocystronic, structural gene monocystronic. Coding and non-coding sequence in eukaryotes, structural gene monocystronic. Structural gene वस्तूंचा monocystronic. Only form one type of monocystronic. Okay. Only form one type of polypeptide. In structural gene monocystronic. Only form one type of polypeptide. Only form one type of polypeptide. Structural gene monocystronic only form one type of polypeptide. एउटा मात्रै टाइपको polypeptide फर्म गर्छ, सिस्ट्रोन. Functional gene sequence, सिस्ट्रोन, TRON, सिस्ट्रोन. Functional gene sequence, functional, FUNCTIONL, functional gene sequence, 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 functional gene sequence. Structural gene monocystronic only form one type of polypeptide. Only form one type of polypeptide. Cistrone, functional gene sequence. Cistrone is a functional gene sequence. Gene co-sequence. Okay. In prokaryotes, structural gene polycistronic. Structural gene polycistronic. Polycistronic, TRONIC, polycistronic. Structural gene forms more than one polypeptide. Forms more than one type of polypeptide. In prokaryotes, structural gene polycistronic. In prokaryotes, structural gene polycistronic forms more than one type of polypeptide. Monocistrone consists of two types of segment in structural gene. Structural gene polycistronic forms more than one type of polypeptide. Functional gene sequence, cistrone. Monocistrone consists of two types of segment in a structural gene. Exon and intron. Monocistrone consists of, okay. Monocistrone, CISTRN, cistrone. Consists of two types of segment in structural gene. consists two types of segment in structural gene. Consists two types types consist of two types of segment in structural gene. Consists two types of segment segment in structural structural gene. Consists two types of segment in structural gene. Structural gene consists two types of segment in structural gene. Monocistron मा दुईटा टाइपको सेगमेन्ट हुन्छ स्ट्रक्चरल जिनमा. exon and intron INTR अस्ताक फेरल्ला exon and intron exon express cistron. Exon express intron intervening ancestron express cistron. Exon and intron भएन. Exon को दुईटा पार्ट express र cistron. Segment which express coding. Intron को intervening र cistron. Segment which does not express interference. Segment which does not express interference. Segment which express coding. Express and cistron. Exon express cistron. Intron intervening cistron. Exon express cistron. Intron intervening intervening cistron. Astaghfirullah. Prokaryotic transcription. Express cistron. Segment which express coding. Exon is segment which express exon. Segment which express 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 coding. Intron intervening cistron. Segment which does not express interference. Segment which does not express segment which does not express interference. FERENC. Interference. Okay. Monocistronic consists of two types of segment in structural gene. Exon and intron. Exon express cistron. Segment which express coding. Intron or cistron intervening segment which does not express but interferes. Template and non-template strand of transcription unit. इसको इतना ही छोड़ते हैं, यहाँ से स्टार्ट करते हैं. template and non-template strand, TEMP, template and non-template, hey wait, it's 14, it's 15, here it is 16, okay, 17, this one is 18. Template and non-template, TEMP, LAT, non-template, template and non-template strand of transcription unit, template and non-template strand. Template and non-template strand of transcription. Transcription unit. Template and non-template strand of transcription unit, okay? Promoter, terminator, structural genes. 3' to 5', 3' to 5', and again 5' to 3'. 5' to 3'. Promoter, terminator, template strand. Okay. Here we have promoter. We are a promoter. Okay. Then we also have terminator here. This is terminator. Three prime to 5', 5' to 3', okay. 5' to 3' like even this downstream and this upstream. 3' to 5', upstream. Template strand, structural gene. Okay, like in this STRUCTURAL, structural gene, GENE, gene. Okay, keep in this yanira banda kera chini. By convention, the other DNA 5' to 3' are taken as reference. Promoter region are present at 5' end of DNA. Euta template strand, euta kita coding strand. Coding DNA at 3' end. Non-template autosense coding strand. I hodgepodge करके दिखाया मैंने इसमें. या अल्लाह. Upstream, downstream. एक बार यहाँ से पढ़ो तो था. RNA polymerase, like DNA polymerase, have 5' to 3' activity. Excuse me. Mechanism of transcription. Enzyme involves DNA-dependent RNA polymerase. That is replication. DNA-dependent DNA polymerase, but here we have DNA-dependent RNA polymerase. DNA template RNA polymerase. Like DNA polymerase, the enzyme also requires the short fragment to continue the polymerization, RNA polymerase. But it can also show de novo synthesis. DNA. RNA polymerase observed three types of RNA polymerase. Again, तीन type DNA polymerase. DNA polymerase 1, DNA polymerase 2, and DNA polymerase 3. यानी रखिएगा 3 types of RNA polymerase observed eukaryote. RNA polymerase 1, RNA polymerase 2 and RNA polymerase 3. 5.8 SR single-stranded ribosomal RNA. 18 single-stranded ribosomal RNA and 28 single-stranded ribosomal RNA. HNRNA, heterogeneous nuclear RNA. 5S ribosomal RNA, tRNA and small nuclear RNA, snRNA. In prokaryotes, RNA polymerase made up of spatial subunit A to beta prime, क्या omega and alpha sigma unit, core RNA plus alpha polymerase, holoenzyme, prokaryote transcription, initiation, elongation and termination. Okay. A transcription unit, a transcription, the process of copying genetic formation from one strand of DNA into the process of copying genetic material, genetic formation from one strand of the DNA into RNA is termed as transcription. Process of copying genetic information from one strand of DNA into RNA is termed as transcription. Here also the principle of complementarity governs the process of transcription, except the adenosine now forms base pair with uracil instead of thymine. However, unlike in the process of replication, which once set in, the total DNA of the organism gets duplicated. Total DNA of an organism duplicated. In transcription, only one segment of DNA and only one of the strands is copied into RNA. This necessitates defining the boundaries that would demarcate the region and the strand of DNA that would be transcribed. Why both the strands are not copied during transcription has the simple answer. First, if both strands act as template, they would code for RNA molecule with a different sequence. Remember, complementarity does not mean identical. They would code for RNA molecule with different sequence. Okay, they would code RNA molecule with a different sequence, and in turn they code for proteins. The sequence of amino acid in the protein would be different. Hence, one segment of the DNA would be coding for one segment of the DNA would be coding for two different proteins, and this would complicate the genetic information transfer machinery. Second, the two RNA molecule, if produced simultaneously, would be complementary to each other, hence would form a double-stranded RNA. This would prevent the RNA from being translated into protein, and the exercise of transcription would become a futile one, pointless. This would prevent RNA from being translated into protein, and exercise of the transcription would become a futile one. Transcription unit. Transcription unit. A transcription unit in DNA is defined primarily by the three regions in the DNA. The transcription unit in DNA is defined primarily by the three regions in the DNA: a promoter, the structural gene, and a terminator. Promoter, structural gene, and terminator. Promoter, structural gene, and terminator. There is a convention in defining the two strands of DNA. There is a convention in defining the two strands of the DNA in the structural gene of a transcription unit. There is a convention in defining the two strands of a DNA in the structural gene of a transcription unit. Since the two strands have opposite polarity, and the DNA-dependent RNA polymerase also catalyze the polymerization in one direction, that is 5' to 3', the strand that has the polarity 3' to 5', 3' to 5', act as template. It acts as template. Okay, यहाँ पर लिखा है ना ये चीज़, मैंने कहीं तो पढ़ा था. यो RNA polymerase like DNA polymerase have 5' to 3' activity. and the strand that has the polarity 3' to 5' act as template. 3' to 5' act as template strand. Okay, यही पर लिखा है, तुम भी लिख दो यहाँ पर. 3' to 5' direction वाला strand, 3' to 5' strand, and the strand that has the polarity 3' to 5' act as template. 3' to 5' strand, strand strand act as template. template, plate, template, the strand as strand, astagfirullah, template strand. The other strand which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil. सेकेंड स्लॉट कंप्लीट हो गया, मैं लिख देती हूँ ना. Three' to 5' strand act as template DNA, whereas 5' to 3', 5' to 3' coding DNA. Coding DNA. Yes, coding DNA. CODING, coding strand or coding DNA. Coding strand, okay. DNA dependent RNA polymerase also catalyze the polymerization in one direction, that is 5' to 3'. The strand that has the polarity 3' to 5', okay, 3' to 5' act as template and is also referred to as template strand. Template strand act as template. The other strand which has the polarity 5' to 3', the other strand which has the polarity 5' to 3' and the sequence same as RNA, 5' to 3', RNA sequence, okay. Five prime to 3', 5' to 3'. This is RNA. RNA, newly synthesized. Alhamdulillah. The other strand which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil. Except thymine. Okay, the other strand which has the polarity 5' to 3' and the sequence same as RNA except thymine at the place of uracil is displaced during transcription. The other strand, which has the polarity 5' to 3' and the sequence same as RNA, is displaced during transcription. Strangely, the strand which does not code for anything is referred to as coding strand. Coding strand, इसको क्या बोलते हैं? Coding strand. STRAND strand. Coding strand does not code for anything. For anything. Astaghfirullah. Coding strand 5' to 3' polarity. Okay. Coding strand removed the sequence, okay, same, is displaced during transcription. To explain the point, a hypothetical sequence from a transcriptional unit is represented below. 3' to 5' template DNA. Okay, coding DNA 5' to 3'. Can you now write the sequence of RNA transcribed from the above DNA? Okay, A को जगह में A U A U, T को जगह में A, G को जगह में C, C को जगह में G, U A C, U A C G, A को जगह में U, U A C, UAC, GTA, GTA, UA, okay, something like this. Template strand, coding strand, same as coding strand on the result. This my U on the result. पर लिखते हैं ना, A को जगह में U, T को जगह में A, C G, U A C G, U A C G, again U A C G, same to U A C G, 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 U A C G. This is the sequence of RNA. Sequence of RNA. Promoter, terminator. A 5' to 3' direction. 5' to 3' direction. RNA polymerase लिखनी ते direction में transcription. की कर log it as answer. The promoter and the terminator flank the structural gene. मैंने वो नहीं लिखा क्या? Structural gene आया ही नहीं, कैसे लिखोगे? Exon intron. Terminator site reason. Terminator site or reason terminates the transcription. Needs the transcription. Transcription, CRIPTION, transcription, right? For prokaryotes, no box, and for eukaryotes, Hogness box में छोड़ो. Promoter side है ना, coding and non-coding sequence आएगी, कर दिया मैंने direct. Structural gene flanked by promoter. Flanked by promoter and terminator, structural gene represents the segment of DNA which undergo conversion. Represent the segment of DNA. Segment of DNA promoted. As for promoter and terminator. It's okay. Express cistron, intervening cistron, segment which does not express interference, okay, coding. Template को direction 3' to 5' हुन्छ template को 3' to 5'. Template strand को 3' to 5' direction act as template for synthesis of mRNA. 5' to 3' वालालाई कोडिङ भन्छ, तो यसले केही पनि कोड गर्दैन, 5' to 3' डाइरेक्सन हुन्छ. Template and non-template strand of transcription unit. Template strand upstream, downstream. Non-template autosense coding strand. Non-template PLAT, non-template autosense, AUTO autosense, SENSE, non-template autosense coding strand. Coding strand. By convention, promoter reasons are by convention, okay, promoter reason are that at 5' end of DNA, 5' end of DNA, क्या बोल रही हूँ मैं? DNA को 5' end में जोड़ सकते लगे इनसे सा promoter. The promoter and terminator flank the structural gene in the transcription unit. The promoter and terminator flank the structural gene in a transcription unit. Transcription unit ma promoter ra terminator structural gene ma huncha. The promoter is said to be located at 5' end upstream. The promoter is said to be located 5' end of upstream of the structural gene, 5' end upstream. Promoter upstream, 5' end upstream, UPSTREAM coding strand. Okay, this is terminator. Three prime. 5' end of 5' end upstream of the structural gene. The reference is made with respect to the polarity of coding strand. Okay, it is a DNA sequence that provide binding site for RNA polymerase, and it is the presence of a promoter in a transcription unit that also defines the template and coding strand. By switching its position with terminator, the definition of coding and template strand would be reversed. The terminator, I think मुझे किसी चीज़ ने काटा. फिर मैंने पता नहीं क्या किया खुद के उंगली को। It is the DNA sequence that provide binding site for RNA polymerase, and it is the presence of a promoter in the transcription unit that also defines the template and coding strand. By switching its position with terminator, the definition of coding and template strand would be reversed. The terminator is located towards 3' end downstream. Upstream and downstream with respect to upstream, STREM, stream, 5' end, and downstream, stream, 3' end, are selected on the basis of coding strand, on the basis of polarity of coding strand, CODING, coding strand. Coding strand को 5' end लाई upstream बन्छ, coding end को 3' end लाई downstream बन्छ. It is a DNA sequence that provide binding. It is a DNA sequence that provide binding site for RNA polymerase. It is DNA sequence that provide binding site for RNA polymerase. It is the presence of a promoter in the transcription unit that also defines in a DNA sequence. It is a DNA sequence that provide binding site for RNA polymerase. And it is the presence of a promoter in transcription unit. It is the presence of a promoter in transcription unit that also defines the template and coding strand. By switching the position, the terminator with the terminator, the definition of coding and template strand would be reversed. And is usually defines the end of the process of transcription. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. Some of the properties of the sequence shall be discussed while dealing with the regulation of gene expression. There are additional regulatory sequences that may be present further upstream or downstream to the promoter. I think था पहले से ही. With terminator, by switching the position of terminator, by switching the position of promoter with terminator, by switching its position with terminator, by switching SWIT, switching, by switching the position of terminator, nator and promoter. It is a DNA sequence that provides binding site for RNA polymerase, and it is the presence of a promoter in the transcription unit that also defines the template and coding. It is the presence of the promoter in the transcription unit that also defines the template and coding strand. Defines coding and template strand. It is the presence of a promoter in transcription unit that also defines the template and coding and template strand. Template, PLAT, template strand, STRAND, strand. Template strand 3' to 5' ones, okay. 3' to 5' with respect to coding strand. Defines coding and template strand. 3' to 5' strand. Defines coding and template strand. By switching the position of terminator and promoter, keep on your reverse answer. The definition of coding and template strand could be reversed. Template and coding strand could be reversed. Transcription unit and the gene. A gene is defined as the functional unit of inheritance. Ambiguity that the genes are located on DNA. DNA sequence. Template and non-template strand of transcription unit. Promoter, terminator, template strand, coding strand. Okay, at 3' end, terminator. Add 5' upstream. Upstream 5' end upstream, 5' end upstream, 3' end downstream. By convention, the other DNA 5' to 3' are taken as reference. By convention, the other DNA 5' to 3' taken as reference. Promoter region are present at 5' end of the DNA. Promoter region coding DNA at 3' end. Terminator, okay. Coding strand, non-template, autosense. Template strand. GAG, CGC, CUC, GCG, CTC, GCG. By convention, promoter region are present at 5' end of DNA. By convention, the other DNA 5' to 3'. Act as reference 5' to 3. reference. Okay, coding reference, coding strand reference को लगी हो all the reference point. Reference point. All the reference point while defining a transcription unit is made with coding. All the reference point while defining transcription unit is made with coding strand. Convention. By convention of the DNA. Coding strand. A reference point while defining transcription unit will be made. All the reference point while defining a transcription. All the reference point while defining a transcription unit is made with coding strand. By convention, the other DNA 5' to 3' are taken as reference. Reference point, point, point taken as reference. Reference point taken as reference. Reference point. By convention, the other DNA 5' to 3'. The reference point. Oh, ये होता था. I'll take a break. Excuse me.
दिदीले ज्वरो नाम नाप्नु त नाम भनेको कहाँ उतार्छ ज्वरो नाप्ने ममा हिजो ज्वरो नापेछ रहेछ ज्वरो नाप्छ त नापेर मलाई भन त कति छ ज्वरो नापेर भन्न त कस्तो छ म यो गरौँ नानी इन्टरनेट अन गर्नु त तपाईँको मोबाइलको तपाईँको नम्बर बताएको छ कस्तो पाराले त्यो भन्दाखेरि छ उठ्नु भन्दा अब 6:30 अलरेडी 6:30 भन्दा स्कुल जानु ढिलो हुन्छ भन्नु त्यति स्कुल जान्छ कि जाँदैन पापाको काखमै पापाको मुखी गरिबस्नु छैन जुन नाप्ने आयो अन गर्नु अन गरेर ज्वरो नापिसकेको नानी यो सुइयो चाहिँ ज्वरो नापिसकेर चाहिँ अफ गरेर चाहिँ गर्नुपर्छ बन्द गरेर कभर लगाएर चाहिँ अलिकति नपुगेर दिदी नपुगेर मात्रै ल्याएको नि खै कहिले सक्दैन रहेछ हौ दिदी सक्छ सकिहाल्छ जति ल्याउँछ नि सकिहाल्छ जति ल्याउँछ सकिहाल्छ त्यो घरले सामान चाहिँ सानो खाइसकेन अब चाहिँ पुग्यो है भन्नु फेरि एकति भ्याक्सिन 100 भ्याक्सिन मगाइदियो भने 100ले पनि पुग्दैन होला हौ सिमेन्ट मगाए फेरि बल्लले पुग्छ होला भने फेरि पुग्दैन फेरि मगायो हो इटाले पुग्छ होला भने इटाले फेरि मगायो अस्ति नै पुरो ल्याएको भयो भने फेरि बेसी ल्याएर पनि के गर्नु फेरि जान्छ भने नल्याएको रे आज त जानु मेला दिदी आज पनि जानु पर्छ होला कहिले
RNA polymerase like DNA polymerase have 3 activity mechanism of transcription. Excuse me. RNA polymerase like DNA polymerase RNA polymerase like DNA polymerase have 5' to 3' activity. DNA polymerase have 5' to 3' activity. Mechanism of transcription.
बोल्दै मासु होइन मासु नै गर्छ नानी एनो ओ लो आयो लो आयो कि दाजु
RNA polymerase like DNA polymerase have 5' to 3' activity mechanism of transcription. Enzyme involved DNA dependent RNA polymerase DNA template. DNA dependent RNA polymerase. RNA polymerase. Enzyme involved RNA polymerase. DNA template. DNA dependent involved. DNA dependent. RNA polymerase DNA template. Okay, enzyme involved RNA polymerase DNA template mounts a DNA dependent RNA polymerase. DNA dependent RNA polymerase. Like DNA polymerase, the enzyme also requires the short fragment to continue the polymerization, RNA polymerase, but it can also show de novo synthesis. Like DNA polymerase, the enzyme also requires the short fragment to continue. Like DNA polymerase, the enzyme also requires the short fragment to like DNA polymerase, the enzyme also requires the short fragment to continue. Like DNA polymerase, the enzyme also requires the short fragment to continue the polymerization. RNA polymerase, but it can show de novo synthesis. Like DNA polymerase, the enzyme also requires the short fragment to continue the polymerization. RNA polymerase. But it can show de novo synthesis. RNA polymerase. Show de novo synthesis. RNA polymerase observed. Polymerase 1, rRNA. 5.8, 18 and 28. Ribosomal RNA. अस्तगफिरुल्लाह, छोड़ो, क्या ही कर सकती हो तुम। 5.8 single stranded RNA, 18 single stranded RNA and 28 stranded RNA. Five point eight. RNA polymerase 2. HNRNA, heterogeneous nuclear RNA. HNRNA, heterogeneous nuclear RNA. Heterogeneous genus nuclear RNA. Heterogeneous nuclear RNA. पागल लड़की. 5s RNA, tRNA, small nuclear RNA, snRNA. 5s RNA कुन कुन छ भन त. 5.8 छ, 18s RNA छ, 28 छ हो यहाँनिर. 5s RNA, 5s RNA, then tRNA, transfer RNA, tRNA, tRNA, snRNA, snRNA, small nuclear, NuCl, small nuclear RNA, okay. tRNA, 5s RNA, tRNA and small nuclear RNA, okay. RNA polymerase 1, 5.8 sRNA, ribosomal RNA, sRNA. 18 and 28 single stranded ribosomal RNA. RNA polymerase 2, HNRNA, that is heterogeneous nuclear RNA, HNRNA, heterogeneous nuclear RNA, HN, heterogeneous nuclear RNA. RNA polymerase 3, RNA polymerase 3, 5 single stranded ribosomal RNA, then snRNA, small nuclear ribosomal RNA कि RNA मात्रै RNA मात्रै छ, snRNA र transfer RNA, tRNA. In prokaryotes 5.8 RNA. In prokaryotes, RNA polymerase made up of special subunit. In prokaryotes, RNA polymerase made up of special A. No, it's 19. In prokaryotes, RNA polymerase made up of special subunit A2, B, B dash, omega, alpha, 4 RNA plus alpha polymerase, holoenzyme. Prokaryotes transcription. Okay, यहाँ से यहाँ तक ना छोड़ दो तुम. In prokaryotes, RNA polymerase made up of special subunit A2, beta, beta dash, omega, sigma unit, core RNA plus alpha polymerase, holoenzyme. Prokaryotic transcription. Prokaryotic transcription, okay. Prokaryotic transcription involves three steps: initiation, elongation, and termination. Prokaryotic transcription involves 3 steps: initiation, elongation, and termination. Okay. Prokaryotic transcription involves involves 3 steps: initiation, yeah. Initiation, elongation, initiation, first 3 steps, okay. Initiation, elongation, and termination. Prokaryotic transcription involve 3 steps: initiation, elongation, and termination. Initiation, elongation, and termination. Initiation: to observe when a subunit of RNA polymerase association recognized bind with promoter is observed. Initiation is observed when a subunit of RNA polymerase associate and recognize or bind with promoter. Let's see. Initiation. First step, initiation. Prokaryote transcription. Okay, you prokaryotes को लगी मैं transcription. Initiation is observed. Initiation is OBSE or VD. Initiation is observed when a subunit of RNA polymerism polymerase when a subunit of RNA polymerase association recognize polymerase recognize association binds the promoter. Initiation is observed when a subunit RNA polymerase association recognizes or binds with promoter site. Terminator, elongation is observed. Initiation is observed when a subunit of RNA polymerase initiates transcription. RNA polymerase binds to promoter and initiates transcription. And initiates transcription. TIN and initiates transcription. Okay. 5' to 3'. 3' to 5'. Promoter RNA polymerase sigma factor. Three prime to 5', 5' to 3'. Promoter site, yellow color with RNA polymerase sigma factor. There is a single DNA dependent RNA polymerase that catalyzes transcription of all of RNA in bacteria. DNA dependent RNA polymerase that catalyzes DNA RNA polymerase binds to promoter and initiate transcription initiation. ना गंदा चैप्टर साला. सिग्मा फैक्टर हो कि ना अल्फा ले पाई मतलब यही लाग्यो हो बता. RNA polymerase sigma factor promoter. RNA polymerase. ये क्या है? सिग्मा फैक्टर. सिग्मा फैक्टर आरएनए पॉलीमरेज. इतना गुस्सा आता है ना, फिर भी पढ़ना पड़ता है. सिग्मा फैक्टर आरएनए पॉलीमरेज सिग्मा फैक्टर मुझे पढ़ना ही नहीं है. सिग्मा फैक्टर टर्मिनेटर इस साइड पे है ना. डीएनए हेलिक्स. promoter terminator alpha factor promoter side RNA polymerase sigma factor. When initiation is observed when a subunit of RNA polymerase association recognized binds the promoter. Involves 3 steps: elongation, initiation, elongation and termination. ये तो मुझे बिल्कुल पसंद नहीं आ रहा है ये चैप्टर. elongation. Polymerization of mRNA with the help of RNA polymerase. Elongation, polymerization of mRNA with the help of RNA polymerase. RNA polymerase is the only RNA polymerase is capable of elongation. RNA polymerase, mRNA. mRNA with the help of RNA polymerase. Polymerization of messenger RNA with the help of RNA polymerase with the help of RNA polymerase. RNA polymerase ले help गर्छ के mRNA फर्म गर्नमा प्रमोटर साइटसँग RNA polymerase बाइन्ड हुनुपर्छ फर्स्ट RNA polymerase is capable of elongation. Polymerase is only capable of elongation. RNA polymerase, mRNA, okay. Elongation. RNA polymerase is only capable of elongation. RNA polymerase capable of elongation. Capable of elongation. RNA polymerase, mRNA. Okay. Termination. RNA polymerase associates with Rho subunit for the process of termination. For the process of termination, Rho subunit recognize the terminator site. And RNA polymerase associated with Rho factor. Rho factor for the process of termination. Rho subunit recognizes the terminator. the terminator. mRNA falls off the row subunit recognizes the terminator. Later, messenger RNA falls off detachment from RNA polymerase. Detachment from RNA polymerase. Detachment from RNA polymerase. Synthesis independent. Synthesis independent. The mRNA falls off. Synthesis independent mRNA. Independent mRNA messenger RNA. Synthesis independent mRNA. Detachment from RNA polymerase. Synthesis independent. Synthesis independent mRNA. Synthesis independent mRNA. Later mRNA falls off detachment from mRNA. Later mRNA falls off detachment from Later mRNA falls off detachment from RNA polymerase. Synthesis independent mRNA. यो अङ्कुलमा चाहिँ के साह्रो हल्ला खल्ला हुँदो रहेछ पहिला ट्याम्के ट्याम पिटिदिनुहोस् नि किन नक्कल पारेको नानीहरु पनि कति जन्माउनु सकेको terminator or termination. Termination. RNA polymerase will associated with the rho factor for the process of termination. Rho subunit recognize the terminator. Terminator site. Rho subunit recognizes the terminator site. Rho subunit recognizes the terminator. Rho subunit recognizes the terminator site. Later mRNA falls off detachment from RNA polymerase. Detachment from RNA polymerase. Synthesis independent mRNA. Synthesis of independent mRNA. In prokaryotes, messenger RNA form does not need any processing as no exon and intron sequence are present. In prokaryotes, messenger RNA form does not need any processing as there is no exon and intron sequence are present. In prokaryote, transcription is exposed to cytoplasm. As there is no breeding distribution. As there is no breeding distribution between nucleus and cytoplasm. Even translation is coupled with the transcription. Wow. prokaryotes. Eukaryotes in prokaryotes. mRNA does not need any processing, as no exon mRNA does not need any processing, as no exon, exon, as no exon and intron sequence are present. वो तो अल्लाह ही जाने, मैं क्या जानूँ. mRNA does not need any processing, as no exon and intron sequence are present. mRNA in prokaryotes. Messenger RNA does not need any processing, as no exon and intron sequence are present. In prokaryotes, transcription is exposed to cytoplasm. Cytoplasm. Operating distribution between nucleus and cytoplasm. Distribution between nucleus and cytoplasm. Translation is coupled with transcription. Translation is coupled with transcription. Translation is coupled with transcription. In prokaryotes, translation is coupled with transcription. आमा शङ्करहरू नेपाली हुन्छ? जसको जात नि शङ्कर हुन्छ तिनीहरू नेपाली हुन्छ? हजुर, मैले पनि सुनेको छुइनँ यस्तो त। أستغفر الله. The mRNA transcribed. أستغفر الله. Let it go, Ima. Let it go. The mRNA transcribed initially are referred as primary transcript. They hold the exon and intron sequence. The mRNA transcribed initially, the messenger RNA transcribed initially are referred as primary transcript. They hold the exon and intron sequence. The mRNA transcribed initially. Are referred as primary transcript. The mRNA transcribed initially. The mRNA transcribed initially are called primary transcript. They contain exon and intron. Primary transcribed initially. The mRNA transcribed initially. Primary transcript. They hold exon and intron sequence. hRNA. Heterogeneous RNA. transcript, primary transcript. Primary transcript RNA, referred as hnRNA. Heterogeneous RNA, referred as heterogeneous RNA. Heterogeneous RNA, primary transcript. Referred as hnRNA, heterogeneous. The mRNA transcribed initially are called primary transcript. Astaghfirullah. The messenger RNA. The mRNA transcribed initially are called primary transcript. They hold exon and intron, referred as hnRNA, heterogeneous RNA, primary transcript RNA, primary transcript RNA. Steps involved in processing of eukaryotic RNA: capping, steps involved in processing of eukaryotic RNA, steps involved in three steps involved in processing of eukaryotic RNA: capping, 7-methylguanine, 7-methylguanosine triphosphate cap is added towards 5' end of primary transcript. This process is catalyzed by guanyl transferase 5' to 3'. Methyl GPPS, guanosine, मिथाइल गुआनोसिन. कितना गुस्सा आ रहा है. ये लोग कितना ज्यादा शोर करते हो यार. एक तो मुझे समझ में ही नहीं आता. एक्सॉन इंट्रॉन, एक्सॉन इंट्रॉन, एक्सॉन इंट्रॉन. It helps in recognizing the smaller subunit of eukaryotic ribose attached with the mRNA. Polyadenylation, adding spliceosome. Complex and removal of introns consist of a small nuclear RNA. Small RNA. Transferase RNA. Genetic अच्छा. Replication and translation. Translation को compare कर रहे हैं. Steps involved in processing of eukaryotic RNA. Capping, tailing, and then splicing. Formation of spliceomes, complex and removal of introns, consist of small nuclear RNA. Types of RNA and the process of transcription. In bacteria, there are three major types of RNA: mRNA, messenger RNA, tRNA, transfer RNA, and rRNA, that is ribosomal RNA. Ribosomal RNA, 80%. All three RNAs are needed to synthesize a protein in a cell. The messenger RNA provides the template. tRNA brings amino acid and reads the genetic code, and the ribosomal RNA plays a structural and catalytic role during translation. There is single DNA dependent RNA polymerase. इतना गुस्सा आ रहा है ना, लेकिन मैं क्या करूं? There is single DNA dependent RNA polymerase that catalyzes transcription of all type of RNA in bacteria. RNA polymerase binds to promoter and initiates transcription. RNA polymerase binds to promoter and initiates transcription. It uses nucleoside triphosphate (NTP), nucleoside triphosphate as a substrate and polymerizes in template-dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix and continuous elongation. Only a short stretch of RNA to the enzyme. Once the polymerase reaches the terminator region, the nascent RNA falls off. So also the RNA polymerase. This results in termination of transcript. It uses nucleoside triphosphate as substrate. Nucleoside triphosphate as substrate and polymerizes in a template. Nucleoside triphosphate (NTP). And polymerizes in a template-dependent fashion following the rule of complementarity. It somehow also facilitates opening of the helix and continuous elongation It's okay, I think. कहाँ गया वो? Okay. And polymerizes in a template-dependent fashion following the rule of complementarity. आगे छोड़ दो. Also the RNA polymerase. 그거 전부입니다. An intriguing question is that how is the RNA polymerase able to catalyze all the three steps, which are initiation, elongation, and termination? The RNA polymerase is only capable of catalyzing the process of elongation. It also transiently associates transiently with the initiation factor sigma and termination factor rho to initiate and terminate the transcription respectively. Association with these factors alter the specificity 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 एक बार में कैसे पढ़ते हैं इस वर्ड को? आई डोंट नो, कोई मेरी हेल्प करना. हाँ, लेकिन अभी कमेंट्स ऑफ है तो तुम लोग नहीं हेल्प कर पाओगे. ऑफ द RNA polymerase to either initiate or terminate. In bacteria, since the mRNA does not require any processing to become active, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in the same compartment in bacteria, since the mRNA does not require any processing to become active, and also since transcription and translation takes place in same compartment, there is no separation of cytosol. And nucleus in bacteria. Many times the translation can begin much before the mRNA is fully transcribed. Consequently, the transcription and translation can be coupled in bacteria. मैंने ये point लिखा है. In eukaryotes, there are two additional complexities. There are at least 3 RNA polymerases in the nucleus. 3 RNA polymerases in the nucleus, in addition to RNA polymerase found in the organelles. There are at least 3 polymerases in the nucleus, in addition to RNA polymerase found in the organelle. There is a clear-cut division of labor. RNA polymerase 1 transcribes. कुछ दिया ही नहीं है इसमें. गुस्सा आता है मेरे को. के खाना आएको थिएन, मलाई पुरा रिस उठेको थियो कि, अनि के गरौँ भएर चाहिँ नि यताउता घुम्नु निस्केको थिएँ. The RNA polymerase 1 transcribes ribosomal RNA. RNA polymerase 1 transcribes rRNAs 28S, 18S, and 5.8S, whereas RNA polymerase 3 is responsible for transcription tRNA, 5 single-stranded RNA, and snRNA. The polymerase 2 transcribes precursor of mRNA, the heterogeneous nuclear RNA, hnRNA. The second complexity is that the primary transcript contain both the exon and the intron and are non-functional. Hence, it is subjected to a process called splicing, where the introns are removed and यहाँ पर कुछ ढंग से लिखा ही नहीं है. It's okay, ma. It's okay. The exon complexity is that the primary transcript contain both exon and intron and are non-functional. Hence, it is subject. हो गया बहन. Hence, it is subjected to a process called splicing, where the introns are removed and exons are joined in a defined order. Okay, splicing. Introns are removed, exons are joined. Where the introns are removed and exons are. मुझे ना 2 गाली देने का दिल कर रहा है, लेकिन नहीं दे सकती. Where the introns are removed and exons are joined in a defined order. Introns are removed and exons are joined in a defined order. hnRNA undergoes additional processing called as capping and tailing. hnRNA undergoes additional process called capping and tailing. In capping, an unusual nucleotide, methylguanosine triphosphate, methylguanosine MGT, methylguanosine triphosphate is added at the 5' end of hnRNA. In tailing, nucleotide, unusual nucleotide methylguanosine triphosphate is added to 5' end of the hnRNA, entailing adenylate residue. 200 to 300 adenylate residue are added to 3' end in a template independent manner. It is the fully processed hnRNA, now called mRNA, that is transported out of the nucleus for translation. The significance of such complexities is now the significance of such complexity is now beginning to be understood. The split gene the split gene arrangement represents probably an ancient features of the genome. Presence of introns is reminiscent of antiquity. And the process of splicing represents the dominance of RNA world. In the recent time, the understanding of RNA and RNA dependent processes in the living system have assumed more importance. अब एक बात बोलूँ, ये ना यहाँ पर जो है नोट्स इस चैप्टर का, ये ना सर ने लिखाया था, समझे? तो नोट्स तो इजी होना चाहिए ना, बिना बुक पढ़े ही समझ में आना चाहिए, लेकिन यहाँ पर जो लिखा है वो समझ में ही नहीं आ रहा है. मुझे इस सर को ना डांटने का दिल कर रहा है. एनसीईआरटी पढ़ने से ही समझ आता है, नोट्स ही समझ में नहीं आता. पता नहीं कैसे लिखा दिया. सर सिर्फ ना मॉड्यूल का ही देते थे. कुछ भी सिंप्लीफाइड नहीं किया है. Eukaryotic transcription in eukaryotes, mRNA formed need processing. mRNA formed need processing. mRNA form need processing. mRNA form need processing. No, to exercise out the non-expressing sequence. mRNA form need processing to exercise out the non-expressing sequence. They hold exon and intron, referred to as hnRNA, primary transcript RNA, heterogeneous RNA. Processing of RNA. Processing of RNA in in eukaryotes. Eukaryotes, processing of RNA in eukaryotes. Okay, capping and tailing. Capping, tailing. The second complexity is that the primary transcript contain both exon and intron and are non-functional. Hence, it is subjected to a process called splicing, where the neutrons are removed and exons are joined in a defined order. HNRNA undergoes additional processing called as capping and tailing. Formation of spliceomes, complex and removal of introns, consist of small nuclear RNA. HNRNA undergoes additional processing called as capping and tailing. Unusual nucleotide, methyl guanosine triphosphate. Unusual nucleotide, unusual circle mark करना था. Unusual nucleotide, methyl guanosine triphosphate. Methyl guanosine triphosphate. Capping of messenger RNA. HNRNA, na primary transcript HNRNA. Capping of HNRNA. Methyl guanosine triphosphate is added in fifth end of HNRNA. Methyl guanosine. Methyl guanosine. Methyl guanosine triphosphate. Methyl guanosine triphosphate. Methyl guanosine triphosphate. Phosphate BHOS. Phosphate. Methyl guanosine phosphate. Methyl guanosine triphosphate is added towards 5' end of primary transcript. Is added. Added to the 5' end of (20, 21, 22, 21, 22. तो Ayo. Methyl of HNRNA. Methyl guanosine triphosphate is added to 5' end of HNRNA. Methyl guanosine triphosphate is added to 5' end of HNRNA. Primary transcript. Transcript, okay. Methyl guanosine triphosphate is added to 5' end of primary transcript. 5' end. This process is catalyzed by guanosine transfer, guanyl transferase. Guanyl transferase. Guanyl transferase. 5' to 3'. Exon intron. Okay, methyl guanosine triphosphate. GTP होता है ना ये, GTP क्या GPP? It helps in recognizing the smaller subunit of eukaryotic ribose attached with the mRNA. अल्लाह प्लीज़ हेल्प मी, मुझे इतना ज़्यादा गुस्सा क्यों आ रहा है? Prime. Guanosine triphosphate. Guanosine triphosphate GTP, okay? Primary transcript. Astaghfirullah. It helps in recognizing the smaller subunit of eukaryotic ribose attached with the mRNA. It helps in recognizing the smaller subunit of eukaryotic ribose attached with the mRNA. Polyadenylation, adding poly AAA amino acid वाला नहीं, पता नहीं क्या है ये, cap. Polyadenylation catalyzed by poly A polymerase, formation of splicing, complex and formation of spliceomes, complex and removal of introns, consist of small nuclear RNA, SNRP, small ribosomal, small nuclear RPS, ribosomal की लिए कोई मिली, ribosome, ribonucleol ये करें तो proteins, SNRPS. Ligase binds with exon mRNA. भैया मुझे तो ट्रांसक्रिप्शन पढ़ते ही ना मौत आ रही है, मुझसे नहीं होगा ये चैप्टर. Polyadenylation adding polyadenylation catalyzed by poly A polymerase, cap. Formation of spliceomes, complex and removal of introns, consist of small nuclear RNA. Comparison between replication and translation. It's okay, ma. Methyl guanosine cap is added. Guanosine triphosphate. Methyl guanosine triphosphate is added to 5' end of primary transcript. 5' end of primary transcript na ko add हुन्छ 5' end पट्टि मिथाइल गुआनोसिन ट्राइफॉस्फेट हुन्छ एड हुन्छ. क्यापिङ भन्छ यसलाई मिथाइल गुआनोसिन ट्राइफॉस्फेट गुआनिल ट्रान्सफरेज भन्छ त्यसलाई के भन्छ गुआनिल ट्रान्सफरेज भन्छ. इट्स ओके मा, इट्स ओके. क्या कर सकते हैं, कुछ नहीं कर सकते. पॉज करो इसको. Formation of spliceomes, complex and removal of introns, consist of a small nuclear RNA. Cap. Polyadenylation, adding splicing where the introns are removed. Okay, exons are joined in a defined order. H and RNA undergoes additional processing called as capping and tailing. Methyl guanosine triphosphate. HNRNA entailing adenyl residue. Adenyl residue. क्या होती है adenyl residue? 200 to 300 is added. भाई साहब. Adenyl residue. Adenylate. Adenylate residue. Adenylate residue. इतना गुस्सा आता है ना. 200 to 300 residue बनी कती वाला. 200 to 300 adenylate residue. Adenylate residue. 200 to 300. Okay, 5 prime end. Template independent manner. In a template independent. Fully processed HNRNA, now called mRNA, that is transported out of the nucleus for translation. Okay, it helps in recognizing it helps in recognizing the smaller subunit of eukaryotic ribose attached with the mRNA. Methyl guanosine triphosphate. Triphosphate helps in recognizing the smaller subunit. Helps in recognizing the smaller subunit of eukaryotic ribose. Attached with the Astaghfirullah. Helps in recognizing the smaller subunit of eukaryotic ribose. Smaller subunit of eukaryotic ribose attached with the mRNA helps in recognizing the smaller subunit of eukaryotic ribose. Polyadenylation. Polyadenylation. Catalyzed by poly A polymerase. Polymerase. Introns are removed and exons are joined. Consist of, spliceomes. Complex formation of spliceomes, complex small nuclear RNA. Formation of spliceomes, SPLI, spliceomes, complex, consist of small nuclear RNA, spliceomes complex, removal of introns, formation of spliceomes. Ligase herbs. फाइनली हो गया ये वाला टॉपिक. identity code, tRNA translation, replication and translation, comparison between replication and translation. Okay, flow model. $29. 29th, 30th. One, two, 1 2 1 2 3 4 1 2 3 4 5 6 1 2 एक दिन मान सकते पनि एक दो एक दुई एक दिन मान छैन एक दुई एक दुई तीन चार बहुत डेंजरस था कहाँ गई मेरे को मैं फिर से भूल गई टाइमर ऑन करना। कब से ना हम लोग पढ़े जा रहे थे और टाइमर ऑन ही नहीं किया। Oxen like IAA and indole butyric acid IBA have been isolated from plants. Naphthalene, acetic acid, and 2,4-D, 2,4-dichlorohydroxy, dichlorophenoxy, not hydroxy, phenoxy, dichlorophenoxyacetic. हम लोगों का फाइनली ना ये 4th स्लॉट कंप्लीट हो गया है। अब मैं ना पूरा लंबा 1 आवर का ब्रेक लूँगी। अरे कुछ भी करूँ कांड ही हो जाता है सारा। Ya Allah, okay.
आई थिंक मैं थोड़ा लेट हो जाऊंगी 5 मिनट जितना.
तिमी आऊ न अन्त म फेरि अहिले नै 1 बजीसम्म पनि खाना खान्छु.
मैंने सुन लिया टाइमर बीप हो गया है, लेकिन तुम लोग पढ़ते रहना। हाँ, मुझे थोड़ा टाइम लगेगा।
पेन ही खत्म हो गया। पेन ही खत्म हो गया। पेन खत्म हो गया, इंक खत्म हो गया। Selena, cell division. Shoots. Growth of lateral and adventitious shoot. Growth of lateral and adventitious shoot. This is cytokinin. Okay, cytokinin. Growth of new leaves. Growth of chloroplast. Delay senescence. Delay leaf senescence. Aging. Delay leaf senescence. Delay aging. Cell division produces new leaves. Cell division produces new leaves, produces chloroplast in new leaves, lateral shoot growth, adventitious shoot growth, and overcome apical dominance, nutrient mobilization, delay senescence in leaf. Cell division produces new leaves, produces chloroplast in new leaves, lateral shoot growth, adventitious shoot growth, overcome apical dominance, nutrient mobilization, delay senescence in leaf. Cell division produces new leaf, produces chloroplast in new leaves, lateral shoot, adventitious shoot growth, overcome apical dominance, nutrient mobilization, delay senescence in leaf. Cytokinin. Ethylene abscess, ethylene and abscisic acid. Cell division produces new leaves, produce chloroplast in new leaves, lateral shoot growth, adventitious shoot growth, overcome apical dominance, nutrient mobilization, delay senescence in leaf. Cell division produces new leaves, produces chloroplast in new leaves, lateral shoot growth, adventitious shoot growth, overcome apical dominance, nutrient mobilization, delay senescence in leaf. Produces new leaves, produces chloroplast in new leaves, lateral shoot growth, adventitious shoot growth, overcome apical dominance, nutrient mobilization, delay senescence in leaf. Cell division. Growth of lateral and adventitious shoot. Growth of lateral and adventitious shoot. Growth of new leaves and chloroplast. Growth of chloroplast. Delay leaf senescence or easing. Delay leaf senescence. Overcome apical dominance, nutrient mobilization and cell division produces new leaves, produces chloroplast in new leaves, produces chloroplast in new leaves, produces new leaves, produces chloroplast in new leaves, lateral shoot growth, adventitious shoot growth, and overcome apical dominance, nutrient mobilization, delay senescence in leaf. Selena. Cell division, growth of lateral and adventitious shoot, growth of lateral and adventitious shoot, growth of new leaves, chloroplast, growth of chloroplast, production of chloroplast produces new leaves, production of chloroplast in new leaves, delay senescence in leaf, delay senescence in leaves, delay senescence. Overcome apical dominance, nutrient mobilization, overcome apical dominance. Cytokinin helps retain chlorophyll. हाय मैं नहीं पढ़ सकती, इतना मुश्किल. Cell division. Cell division. Okay. Growth of growth of lateral and adventitious shoot, lateral and adventitious shoot. New leaves and chloroplast, CHLOR chloroplast production. Delaying. Delay senescence. Overcome apical dominance. Cytokinin. Mobility of nutrition. Mobility of nutrition. There are total 6 functions, right? Cell division, growth of lateral and adventitious shoot, new leaves and chloroplast, production of chloroplast and new leaves, delay senescence, overcome apical dominance, mobility of nutrition. Cytokinin have specific effects on cytokinesis and were discovered as kinetin, a modified form of adenine, a purine, from the autoclaved herring sperm DNA. Cytokinin. Kinetin, a modified form of adenine. Kinetin. From the autoclaved herring sperm DNA. Kinetin does not occur naturally in plants. Search for natural substances like cytokinin with cytokinin-like activities led to isolation of gelatin from corn kernels. Search for natural substances with cytokinin-like activities led to isolation of gelatin from corn kernels. Corn kernels and coconut milk. Zeatin. Corn kernels and coconut milk. Since the discovery of gelatin, several naturally occurring cytokinins and some synthetic compounds with cell division promoting activity have been identified. Natural cytokinins are synthesized in regions where rapid cell division occurs, for example, root apices, developing shoot buds, young fruit, etc. It helps to produce new leaves, chloroplast in leaves, lateral shoot growth, and adventitious shoot formation. Cytokinin help overcome the apical dominance. They promote nutrient mobilization with the help of which helps in delay of leaf senescence. Cytokinin. Cell division, correct. Shoot. Overcome nutrient mobilization, overcome apical dominance, mobility of nutrient mobilization, अबे भाई बाइनरी ऑप्शन चुप कर भाई, मेरा तो 165 आया था तब।